Okay, everyone. So we have talked about the different parts of our digestive system. We have talked about how there are differences in different species, adaptations to our different diets. We talked about the different steps it takes. Now we're going to finish our chapter by talking about how digestion is regulated. So each step in our digestive system is regulated as needed. So, for example, you don't start producing saliva until the nervous reflexes in your mouth recognize that you have food in your mouth, right? So we don't go around drooling on ourselves all day long, hopefully. We are regulating each step so that we're only producing what we need to digest food when we have it. There's a whole division of the nervous system, the enteric division of the nervous system, which helps regulate the digestive process. That nervous system, again, is your brain triggering your nerves to control your body parts very quickly. We also have our endocrine system that also regulates our digestion through hormones, through releasing and transporting hormones. One example of a hormone would be as food enters your stomach, it causes the stomach to stretch, and that releases the hormone Gastrin in the bloodstream. When Gastrin gets to the stomach, it stimulates the production of gastric juice so that you can prepare to digest the food that's reached your stomach. Another hormone would be produced when chyme arrives in the duodenum. Remember where the duodenum is in the small intestine? When chyme gets into the duodenum, it releases a variety of different hormones. One example would be CKK or cholecystokinin. This stimulates the release of pancreatic enzymes and bile from the gallbladder to help digest your food. Secretin causes a release of bicarbonate ions from the pancreas to neutralize stomach acid. And if chyme is rich in fats, those high levels of secretin and CKK will inhibit peristalsis and secretion of gastric juices, which will slow down the digestion so that you're able to digest the food. So here you can see Gastrin releasing gastric juices. You can see where secretin and CKK are activated, and then also how we can inhibit secretin and CKK. Okay, after a surgical removal of an infected gallbladder, a person must be especially careful to restrict dietary intake of what? Starch, protein, sugar, fat, or water. Go back to that last slide to see what gallbladder is important in helping with, and which of these would you need to restrict your dietary intake of if a gallbladder was removed. We're also able to regulate the energy storage that we have stored in our bodies. So if you are eating more calories than your body needs, those extra calories are going to be stored for later use. And people, the way that we first start storing our body energy is first glycogen in the liver. Glycogen is a complex carbohydrate that we store in the liver and in muscle cells. And then if you have about a day's worth of energy stored in that, any additional energy after that is going to be stored as fat in adipose tissue. On the opposite side, if fewer calories are taken in than expended, so maybe you've been exercising a lot and using more calories up, you're not eating as many calories as you're using, the liver will release that glycogen. So that glycogen here that is being stored when you eat too much, when you don't eat as much, the glycogen is released to be an energy source. Released from your liver first and then your muscle cells, and extra energy as fat and adipose tissue is going to be released for your body to have energy. Most of us have enough fat in our bodies to sustain them for several weeks without eating. Most people can go generally at least 10 days without food. It's also really important that we keep a balanced level of glucose in our blood. So that is glucose homeostasis. That is important for our metabolism to be able to function correctly. Normal range would be 70 to 110 milligrams of glucose per 100 milliliters. This is needed to be able to do cellular respiration and also as a carbon source. And we regulate the amount of glucose that's in our blood sugar with two hormones, insulin and glucagon. So we have a negative feedback loop here with these, whether we're releasing insulin or glucagon. Insulin is stimulated by an increased blood sugar. So when you have more sugar than you need in your blood, insulin is going to be released by beta cells in the pancreas. This is going to cause glucose to move into cells, move into the liver cells, and that is going to mean there's less glucose left in the blood. So your blood glucose falls back down to normal. If you have a low blood glucose level, that stimulates glucagon to be released. Glucagon is released by the alpha cells of the pancreas. So these are both released by pancreatic cells, but different kinds of cells here. So glucagon is released by alpha cells in the pancreas, and that triggers glycogen to be broken down from the liver and released into the blood. And so now there will be more glucose in the blood, bringing the blood glucose level back up into the normal range. Okay, we're going to watch a video about this on our next slide.
Hi, this is Kelly Hogan from the University of North Carolina. In this Visualizing the Concept video, we'll walk through how pancreatic hormones regulate blood glucose level. We'll also consider what happens when that regulation goes awry. Let's get started. Your cells need fuel to function, and glucose is an important energy source for almost all cells. What would it look like if you graphed blood glucose level throughout the day? Would it be a straight line? Would it rise and fall? Blood glucose level fluctuates throughout a day, so we would see a wavy line, like this one. Notice that as the glucose level rises and falls throughout a day, it hovers around a set point. The regulation of glucose level around this set point demonstrates an example of homeostasis, which is a state of equilibrium or steady state of body functioning. The blood glucose set point is maintained by two antagonistic hormones, insulin and glucagon. These two pancreatic hormones are said to be antagonistic because the effects of one oppose the effects of the other. Both hormones are always present, but the level of each fluctuates depending on your diet, exercise, and other factors. To begin, let's consider the effect of a meal, such as a carbohydrate-rich cereal breakfast at 7 o'clock in the morning. As breakfast is digested, glucose monomers are readily absorbed into the bloodstream, causing the sharp rise of blood glucose level on our graph. The sharply rising line we just observed on our graph corresponds to what we will now see represented in the blood vessels. After a carbohydrate-rich breakfast at 7 a.m., we see an increase of glucose molecules in the bloodstream. The rising blood glucose level stimulates the pancreas. In response, beta cells of the pancreas secrete insulin that diffuses into the blood. The insulin secreted from the pancreas travels throughout the whole body through the circulatory system. When insulin diffuses out of the blood vessels, it binds to receptors located on the surface of most cell types. Notice that insulin itself does not enter the cell. The binding of insulin to receptors on the outside of the cell initiates a signaling pathway inside the cell. As a result, cells are temporarily able to take up glucose when insulin is present. The glucose will be used as cellular fuel. Some cells, like liver cells, are like a bank. They store excess glucose and release it when it's needed. The binding of insulin to the receptors on liver cells causes them to take up, as well as store, glucose as a polysaccharide called glycogen. Muscle cells store glucose in the form of glycogen too. Actively contracting muscles require large amounts of energy, so storing glucose directly in muscle cells is efficient for when the muscle cells require immediate fuel. Once the various cells of the body have taken glucose in, the once high blood glucose level decreases. We see that here, represented by fewer glucose molecules in the bloodstream. As blood glucose level decreases, the beta cells of the pancreas are no longer stimulated to secrete insulin. Insulin secretion slows. The blood glucose level is now nearing the set point again. Let's summarize what we have just learned with our graph. At 7 o'clock a.m., blood glucose level rises after a carbohydrate-rich meal. insulin is secreted and blood glucose level declines. Thus, insulin production lowers blood glucose level. Let's now examine how the antagonistic hormone glucagon raises blood glucose level. What happens if we go too long without eating, such as if we skip lunch on a busy day? When we go an extended time without food, blood glucose level begins to decline. The declining line we just followed on our graph is represented in the blood vessel here. No food has been eaten since breakfast, and it is now past 2 p.m., resulting in fewer glucose molecules in the bloodstream. The declining blood glucose level stimulates the pancreas. In response, alpha cells of the pancreas release glucagon. As we saw with insulin, glucagon diffuses out of the blood. It binds to receptors located on the surface of liver cells. The binding of glucagon to receptors on liver cells results in the breakdown of glycogen polymers into glucose monomers that are then secreted. The secretion of glucose from liver cells causes the blood level to rise. The glucose is now available for use by all cells of the body. With more glucose molecules in the blood, the alpha cells of the pancreas are no longer stimulated to secrete glucagon. Glucagon secretion slows. The blood glucose level is nearing the set point. We can now explain why the line on our graph declines and rises when a meal is skipped. Going an extended time without eating causes blood glucose level to decline. Glucagon is secreted and blood glucose level increases. If beta cells of the pancreas were absent, which hormone would an individual not be able to produce? And as a result, what would this individual's cells not be able to do? In the pancreas, the beta cells are responsible for insulin secretion. Insulin's presence allows most cells to take up glucose. Thus, if beta cells of the pancreas are absent, an individual would not make the hormone insulin. And without insulin, cells could not take up glucose. With less fuel, they wouldn't be able to complete as much cellular work as normal. In the absence of insulin, what would you predict the blood glucose level to be? Lower than the set point, higher than the set point, or equal to the set point? The blood glucose level would be higher than the set point. Although glucose would be plentiful, without insulin, many cells would not be able to take up glucose, and they would starve. As more and more glucose remained in the blood and not inside cells, blood glucose level would stay elevated. It's normal for blood glucose level to fluctuate around the set point throughout the day. But abnormally elevated blood glucose level is known as hyperglycemia. The serious hormonal disorder, diabetes mellitus, is characterized by hyperglycemia. Did you know that 1 in 12 Americans has diabetes? And by 2050, if current trends continue, 4 out of 12, or 1 in 3 Americans, will be affected. Hyperglycemia associated with untreated diabetes can cause dehydration, blindness, cardiovascular, and kidney disease. With statistics like these, it's imperative we all understand homeostasis of blood glucose.
Okay, so that video ended with talking about diabetes. When cells are unable to take up enough glucose to meet metabolic needs, blood glucose levels may be higher than the kidney's ability to absorb it, and glucose will remain in the blood. It'll also end up with frequently more sugar in the urine than is normal. There's 2 different types of diabetes, type 1 diabetes and type 2 diabetes. Type 1 diabetes is an autoimmune disorder. In this disease, the immune system has created an immune response to the beta cells of the pancreas and starts destroying those cells. This usually appears in childhood, and it can be treated with insulin injection several times a day. Type 2 diabetes is not insulin dependent. It is characterized by a failure of target cells to respond normally to insulin. So you are still making insulin, your cells just don't respond to them. So you can't treat that with an insulin injection because the cells wouldn't respond to that either. Excess body weight and lack of exercise increase the risk of developing type 2 diabetes. And this generally appears after the age 40, typically only in younger people if they're sedentary.
In diabetes, the body is unable to maintain homeostasis of blood glucose levels. In type 1 diabetes, the beta cells of the pancreas are destroyed by the immune system, and so no insulin is produced. In type 2 diabetes, the pancreas produces insulin, but target cells do not take up glucose. In both types of diabetes, when blood glucose levels rise, cells do not take up the additional glucose, and so blood glucose remains high.
We also have hormones that help regulate our feelings of appetite and our feelings of being full. Overnourishment can cause obesity, which is when there's excessive energy stored as fat, and this contributes to several diseases, including type 2 diabetes, breast cancer, colon cancer, heart attacks, and strokes. Our body does have mechanisms to help regulate body weight. There are hormones that regulate appetite by affecting a satiety center in the brain. One of those is Ghrelin. Ghrelin is secreted by stomach wall that triggers feelings of hunger. So this stomach is releasing Ghrelin because it is not having the food in it. Insulin and PYY are secreted by the intestine after meals. Insulin and PYY will suppress appetite and trigger your brain to say, I'm full, I'm satisfied, I don't need any more food. Leptin is produced by adipose tissue. So when fat starts being stored in the adipose tissue, it signals to the brain, I've got plenty of energy, I don't need to eat anymore, and makes that feeling of satiety. So how do PYY and leptin complement each other in regulating body weight? Okay, so that is finally the end of this chapter. This was a big one, but also I think pretty interesting to learn how our bodies regulate something like our need to eat. If you do have any questions, please let me know, and I will talk to you later. Bye.